Hi, I'm James Verdier, and welcome to the American Institute of Biological Sciences BioScience Talks, which is a forum for integrating the life sciences. On the second Wednesday of each month, and sometimes in between, we discuss the latest bioscience publications. And as a reminder, if you'd like to read more, point your browser to academic.oup.com/bioscience. Today's episode is a very special one. I had a chance to chat with 9 members of the biological collections community about biodiversity infrastructure and its role in the present COVID-19 era, both as a means of enabling ongoing education and research and also potentially helping detect and mitigate future threats to human well-being. These topics and others are covered in 2 bioscience publications, an editorial and a viewpoint, that are published in conjunction with this episode today. There are links to those in the show notes. And to keep the intro short, I've placed the guest info in the show notes too, and today's guests introduce themselves throughout the show. And along the way, you might hear some familiar voices, and that's because this episode builds on an earlier discussion of the extended Specimen network, and I recommend that episode to those who'd like to learn more about biodiversity infrastructure. But for now, let's go to the discussion. All right, so first of all, thank you all very much for joining me today.
Thank you. Glad to be here.
Yep,
Nice to
glad
be
to
here.
be here.
Hi.
Thanks.
Okay, to get us started, I was hoping we could talk about collections in the modern sense, just kind of generally. So it's probably a myth that's, too old to even bother dispelling almost. But, we're not necessarily talking about just, an insect pinned to an index card or, an old bone stuffed away in some musty old closet somewhere in the basement of a museum. But what kinds of things are we talking about?
Well, I'll start and just say that actually we are talking about bones in a drawer and pinned insects. And the amazing thing is that with those specimens is all this other information. And one of the things we've got the capacity to do now is take advantage of new technology with respect to making those kinds of data enhanced and more readily available in all kinds of different ways. And that includes research and for just basically data access. And so that's the big thing that we're trying to do right now.
Yeah, and I don't know about bones in a drawer tucked away in a basement somewhere. I think that's a mischaracterization of what collections are. Collections are biodiversity in a museum that allows for the study of biodiversity almost as well as the study of biodiversity in nature itself. So from that perspective, I think it's an old-fashioned thought that somehow these are dusty things all stored away somewhere never to be seen again, because that's certainly not what modern biodiversity science want to do. They want to see these specimens in collections be used for very active research and grand challenge questions for today's science.
And collections aren't old either. That's another myth, that these are all old things. Hundreds of thousands of new collections are made every year and are deposited collections of bones, for sure, of butterflies, but also tissues and hair and eggshells, a whole host of things.
I always like to mention here, it's also the expertise. So not only do you have this network of data, but you have this network of experts that have really led the way in the idea of open and collaborative science, because we've always been working globally. We've always been sharing this information because a lot of the biodiversity we need to look at much bigger scales or timeframes. And that's what sort of the collections kind of pull together by creating this network.
And I'd like to add that collections today are not isolated from one institution to another, but they're connected globally because they're digitally connected. So we have resources around the world that we can access through digitized specimen information. We have images, we have all sorts of information that we can share with people everywhere. All we need is an internet connection and we can tap into the vast resources of our global collections.
Okay, perfect. I think that is an excellent overview. But moving on, I think we might as well come right to one of the topics of our discussion today, and that is the role of collections, and things like the extended specimen network in helping us identify, and mitigate future disease threats, and how this biodiversity information might play into that sort of project. I'll just leave it at that and leave it open and collect your thoughts. And as we do that, why don't you folks go ahead and state your names so that our listeners will have a chance to know who's speaking.
I'm John Bates, and I'm a the current president of the Natural Science Collections Alliance, and I'm also a curator of birds and head of life sciences at the Field Museum of Natural History in Chicago. And what I'd say with respect to this current COVID situation is that it illustrates just how important science is, in the sense that the minute that this virus started being noticed in China, the first thing someone did was sequence its genome. And then the next thing they did was start asking questions about where it came from and what it was related to. And viruses are parasitic in the sense that they live in hosts. And so one of the first things that came up was what kind of hosts was this particular virus living in? And this is information that relies on understanding biodiversity. And this particular case actually has some very interesting questions associated with it in terms of whether it came from things like a pangolin or different types of bats. And each one of those questions has really basic biodiversity aspects to it that can be best answered by some of the material that we have in our collections.
This is Pam Soltis. I'm a curator in the Florida Museum of Natural History at the University of Florida, and I'm part of the iDigBio project. And I think that digitized collections give us access to so much information worldwide that we can use to help address a whole range of biodiversity-related societal problems and challenges that we're facing this century. So these range in topics from how species will be responding to climate change to how we might be able to predict the next global disease outbreak or pandemic. So I think there's a lot of information in our collections that just allows us to, first of all, pair species together with their pathogens. The collections give us information on natural distributions of pathogens and their hosts. And we have access to genetic resources that are also coupled to these distributional, climate, and host specimen information. So if we actually have all of this information linked tightly together through informatics mechanisms, we can start to pair up information about where a virus has been detected, what its hosts are, where we might go to find out more information about the distributions of those hosts, what other interacting species occur with the hosts, how we can get genetic information on those hosts, and ultimately then on additional pathogens. So we have this whole broad range of information that's all available through connected information about different aspects of collections.
Yeah, I agree 100% with that, Pamela. And I think that I'm Gill Nelson, and I'm the director of iDigBio at the Florida Museum of Natural History at the University of Florida. And our job is to digitize specimens and make data available, mobilize it so that it is stored in aggregators and available to researchers across the globe. And when we first started this, we talked about making it easier for people to get to specimens without having to travel all around the country to find the specimens that they wanted to study, and also run the risk of missing specimens because they don't have the finances or resources to make every museum where certain specimens might be held. And so I think what Pam just said is, to me, the most exciting part, and that is the ability now, forget about the travel geographically, now we can begin to combine these data together in new ways, find new relationships all across collections domains, vertebrates, invertebrates, viruses, fungi, and begin to see these relationships without having to actually, I don't think we could do that if we didn't have the ability to digitally provide data sources to folks to look for relationships.
This is Barbie Tears from the New York Botanical Garden. Just to point out that diseases in humans in animals is the topic of the moment, but equally important are the ideas or the possibility of diseases that could destroy major crop plants, could destroy our food or our other important products. And also collections tell us a lot about how species become invasive and how an outside species can move in and force out natural ecosystems, and also things like the right kind of grass for livestock to eat and so forth.
Yeah, and I'll just add to that it's important to remember that basically all these disease related things like bacteria and viruses have been co-evolving with the biodiversity that's out there for millions of years. And so a lot of times they may have already come up with solutions that we're just now beginning to get the tools to even study. And so those kinds of studies may provide answers for us that we haven't even thought of yet. And so this is where being able to go back to the specimen and understand exactly what the relationship is becomes really important.
I think this is where some of our genetic resources come into play as well. So there are some excellent examples of natural history collections that have really extensive genetic resources as well. And it's possible to analyze this host specimen for any sorts of pathogens that might be still present in preserved tissues from that specimen. And so this gives us a chance to look at the distributions of pathogens on spatial and temporal scales, as well as interspecific host relationships. And this is all information that could be used perhaps to understand the source of a disease, the transmission dynamics of that disease, and potentially even possibilities for developing vaccines.
Yeah, that's really fascinating. So, just harping on the disease example for a moment, this would be a case in which if you had everything readily, digitized and and sequenced, of course, and people start showing up in an ER with a disease you haven't seen before, with symptoms you haven't seen before. You go through the first step, as John described, of sequencing the virus, and then if you had everything, kind of easily at hand, through a good extended specimen network, you could then see perhaps where else this disease or similar diseases showed up through, any number of samples that you had around the world, and start to understand it much more quickly. And then, start to make moves to figure out, hey, this periodically shows up in, such and such primate, or, this appears to be something that, has just made a jump from bats. And you can kind of look at things in that way that you wouldn't have access to if you didn't have those resources at hand.
That's true. And then another aspect of that is, let's say it's something that you don't recognize and it doesn't seem to fit into place. One of the other things that we can do is, building on what Gill was saying earlier, is we can find out what gaps we have in our knowledge. And so when something novel comes along, we can begin to say, okay, we know the parameters around the edges. This is where we need to go look to try to figure out something that we really don't understand. And this is something that happens all the time in biodiversity questions.
I think also one of the strengths is really that you have the specimen to go back to. And so you may have that data from somebody who did the genome or looked at the diseases, but now you want to figure out, well, how did that group of bats survive? Or how did that group of organisms survive? It's not ephemeral data. It's something you can go back, validate, and then ask additional questions. And that ability to do iterative science is really one of the strongest parts about the fact that we actually have the specimen.
Following up on Anna's point about expertise between collections, I think one of the things that the latest crisis, the COVID-19 crisis, has given us is an opportunity to build relationships with virologists and others with whom we might not work regularly. And I think building that network of expertise so that we can draw on those folks when needed is very important too.
It's also important to to point out that there's predictive value. Based on the collections we have, we know where they occur, we know the conditions under which they live, and what associations they have now. And then using modeling techniques and algorithms, we can begin to predict where the next problems might come. And that gives that, and especially combining that with our public health colleagues, gives a very powerful tool for protecting against future pandemics of one sort or another.
I think it's important to recognize as well that the problem with zoonotic diseases is that human civilizations are encroaching increasingly on those few remaining ecosystems. And so as human populations get closer and closer to areas where there may be human pathogens that have never actually made the jump into humans, we run the risk of increasing the incidence of these zoonotic diseases. We can look at places where human populations are getting particularly close to certain populations of, let's say, bats, which are known to harbor large numbers of coronaviruses in particular. And so we can start to make these sorts of predictions about where outbreaks might occur, what types of outbreaks there might be, and a whole host of other sorts of incidents.
Yeah, and that would certainly be something that will become only more valuable as people are living in, conditions and closer to ecosystems that only make such transfers more likely over the passage of time. Another thing I was wondering is, what collaborations and alliances are not yet in place that would enable, this sort of work. I of course, funding would always be, better if there were more of it. But, what sort of relationships between different, researcher communities and scientific communities would be helpful in, making some of these things more likely to actually happen in the future, were we to encounter such a happening again at some point?
Well, I like to use this analogy out of the COVID-19 example, which is that the places that dealt with COVID-19 most effectively were places that had actually had SARS viral pandemics before, so places like Singapore and South Korea. And so, I like to use that as evidence that we can actually effectively learn from what's happened in the past and put things in place more effectively. And now that's a health example, but as Gill was talking about, this has opened up an opportunity to talk to the viral community and have the biodiversity community talk to the biodiversity community. And these are, really important things. And I think a lot of times the two communities don't recognize how much, one, they have in common and how much their goals are in common, but two, how synergistic the information that they have is. So, a good example within a group like Rhinolophus bats, which is one of the things that's hypothesized to be a source of the COVID, the current pandemic, is that the taxonomy of those bats has not worked out well. So if you're a virologist and you're trying to make an assumption about a Chinese horseshoe bat and whether or not it's the source, you actually want to, one, have a voucher specimen so that you know that you can look at it. And then two, you want to go and talk to somebody who can tell you that Chinese horseshoe bat is actually a Chinese horseshoe bat and not one of 4 or 5 other possibilities. And this is something that the two communities have to offer one another.
And what's it like forging that sort of collaboration? Speaking only for the lay community, I think we have a tendency to have this notion that there is a science community and that you're all in it, and that you all gather in one Discord chat room and all talk together, which is of course not the case. But what's it like putting together those relationships and finding out whom to talk with in those various settings?
Well, that's a very, very interesting question to me. And I was at a workshop just recently for a parasite tracker program, problems that folks are addressing. And at that meeting, there were a lot of entomologists. And there was also somebody from Walter Reed who heads up or works with the mosquito collections. And Walter Reed, you think, would be at the Walter Reed Hospital, but it's not. I think it's at the Smithsonian or somewhere. And so So it seemed to me that although most of the entomologists knew about the mosquito program, they didn't really know about the mosquito program, and the presentation was super helpful. And I think that comes from the fact that the folks that work at Walter Reed, they have a goal and a procedure. They have some things they're trying to achieve. And I don't know that we always see ourselves, even in collections, in the same community. And so figuring out how to find those folks like you suggest, I think is absolutely essential so that we do spend more time at professional meetings together.
Right. We do love to meet and talk with one another, but there still remain silos. And there is a tendency for people who work in one particular type of collection or a particular type of organism to flock together. I think the last decade, and due in very large part to the effort of iDigBio, we have a much more national, robust collections community than we ever have had before. And so that's a huge step. But, we actually don't know all the collections we have in this country. We don't have an email address for every single person who is responsible for a collection in this community. So we don't even really know the full scope of our own community, let alone all of the institutions, researchers who may be holding derivative data, those so-called extensions, in collections from our specimens, nor do we fully know the full range of people who use our data. We don't have the kind of tracking data about users that would let us know whether we're giving them what they need or whether there's something more we could be doing.
This is also true of conservation biology, where we're not attending conferences of conservation biologists. They are not necessarily attending conferences of botanists and entomologists and others. And so somehow we've got to break that down. I was interested in what Barbara said about sort of building collections community. When iDigBio first started, we were doing workshops for people. And of course, we said, well, let's do one for the botanist. And then, well, now we got to do one for the entomologist. And now we need to do one for the folks who store things in wet collections. And everybody thought, well, that's fish. And so after that third workshop, I had a fellow call me over and say, you missed the boat. I said, what do you mean? He said, I've got huge cabinets full of insects stored in fluid. And you didn't talk about that during this workshop. And following that, we decided we were on the wrong path. And we began to try to have cross-domain workshops for most of what we did, so that people from various ranges came. And then as they shared what they were doing, other domains could say, hey, we can use that technique ourselves for whatever research or digitization effort or storage effort that there might be.
Yeah, if I could just build on that, I think one of the things we're seeing is the result of these kinds of efforts. And one of the, you can't get away from funding with respect to the differences in interacting with the health community versus the biodiversity community, because we are funded for the most part in very different ways at the government level and even elsewhere. But I think that the capacity to put these data sets together that's happening as a result of these projects like iDigBio is actually creating the synergies to do the kind of science that actually integrates across different types of different groups. And so in a situation where in general over time we've all been experts in our individual areas, it's now much more possible to reach out to a collaborator who works on another group and say, hey, let's look at this. And NSF is, to their credit, has actually been responsible for funding of this, a lot of this through this ADBC program that allowed for digitization of things like parasites, for instance, which are being tied to their hosts across these collection databases in ways that we think we'll learn a lot from.
I think extended specimen network concept is pushing that even further, so that the cross-fertilization between all of these domains is going to grow and be very helpful.
So, James, I have to do a shout out to this, but I'm Anna Monfils. I'm at Central Michigan University, and I'm PI on the Blue Project, which is looking at getting sort of these databases that are emerging, these biodiversity databases, into our undergraduate curriculum. And so the reason I'm bringing this up right now is because, yes, there's the issue of trying to reintegrate biology that's definitely being dealt with and we're looking at, and it's really critical. But we're also still training the next generation of scientists. And one of the ways to reach those future medical doctors and future conservation biologists with this data and the potential of this data is by introducing it early and often as a resource, just like all the other resources they've gotten used to. You know, they know about GenBank, they know, but are they equipped with the skills, and do they understand the data coming out of the biodiversity data sets in a way that they can apply it? And so I think that's one mechanism too that there's been some important energy behind as well.
Yeah, absolutely, and I think that's a fantastic segue. If you ever want to host a podcast, you certainly have the skills. Because I think now is also a great time to talk about education and the potential for collections to act as a learning tool as well. So kind of what's that effort been like, and is it is the notion to just ensure that you bring these resources to bear for people early on in their academic careers, so that then they're aware of them and they know how to use them, regardless of where they end up, or is this mostly exposure for future collections folks, or are we talking about those who might go into the biomedical sciences as well?
That's an excellent question. And so several years ago, I can't really remember, it might have been 2014, Pam and I were involved in something at the Florida Museum where we picked out 15 students that were biology students from universities in the state of Florida. These were not high school kids. These were kids who knew they were interested in biology. We brought all 15 of them to the University of Florida, to the Florida Museum for a couple days. They went through all kinds of things in the back of the museum. They extracted DNA. They did all kinds of good stuff. And it turned out that of those 15 kids, not one of them knew about collections in museums and the research that was going on with those collections. And I thought, man, we have a lot to do in undergraduate biology education for students to learn about collections.
Yeah, absolutely. And this kind of almost gets back to that COVID discussion a little bit, but obviously everyone has had to make enormous changes to the way that they're teaching. And that has, precluded a lot of undergraduate experiences doing research, et cetera, that otherwise, would be available. But the collections are still there and they are still accessible, through various digital means. So what kinds of things have been going on that have allowed students to continue doing real work and real research and to learn in that fashion?
Sure. I'm Janice Krumm. I'm at Widener University, and I am the PI on BCEENET, which is Biological Collections in Ecology and Evolution Network. So BCEENET has recently received a rapid grant, and what we are doing is this summer, we're actually in the process of, with teams of both collections professionals and undergraduate educators, designing and creating the materials to be able to put course-based undergraduate research experiences using the digitized collections in place for fall semester and spring semester this year. One of the great tragedies of COVID as an undergraduate educator has been with the students not on campus, and that being a question mark moving forward in the fall. Research experiences have been lost in many cases or inaccessible, and being able to bring the research into the classroom, into their coursework, is a way to expand opportunities, especially for students who, for whatever reason, be it they can't get to campus because of health issues or family issues or whatever, but to make the opportunities available for them to actually get research experience. And the collections are especially useful for this because all of the students, whether they're at a community college or an R1 institution, they have access to those same resources. And so it levels the playing fields in ways that are not possible in some other fields. And so we're really excited about we've had a tremendous response from educators and collections professionals, and an enthusiasm about this We were shooting for, I think, a dozen colleges, and it's going to be potentially much larger response than that.
So I'm a co-PI on a thematic collections network grant through this ADBC program that's called OVER. And the goal of that project is to CT scan as much of representation of vertebrate diversity as we can over the course of the project. And if you think about it, this was started because it's going to be a great research tool for everybody, but all the information is deposited virtually and accessible via a common data portal called Morphosource, which is also NSF funded, and it's tied to real specimens. But the benefit of having extended specimens like these CT scan specimens available via Morphosource is that they're accessible as teaching tools for anybody at any given time, provided they have the skills to work with the data. And now that's a steep learning curve. But I think one of the things we're finding as a community is we need to come up with the ways to make those tools more accessible to students, and that'll make accessible stuff remotely that we never even thought about even 10 years ago.
And this is, I'm Liz Shea, and I'm the curator of mollusks at the Delaware Museum of Natural History. And I've been working with Janice Krumm and Carly Jordan on the BCEENET activities. And one of the things, I'm not an educator. I mean, I am, I love education and I participate whenever I can, but I don't have students of my own. I fall squarely in the collections professional end of things. But these sorts of collaborations are really so very useful, even for somebody like myself who doesn't have students, because it actually helps the collection. Not only does it meet the mission of our museum to explore and discover, it gets students involved in it, but it also improves the collection. And so really it's a win-win for us. It's great to be interacting with the students, but also to get additional georeferencing done, or to get students excited about taxonomy and systematics. That has a follow-on effect that is only positive. And so it's been a lot of fun getting involved in these sorts of things.
Yeah, and it's bringing not only the students into the collection, but it's also bringing educators into the collection that otherwise would never have imagined being able to access any of these resources. I think we have a lot of work to do in spreading the word and making sure that educators that aren't just going to the big scientific conferences know about this, but at all levels of institutions are aware that it's available and it's accessible, and the tools to get them started in it are available. And I think one of the great resources that also is out there is Blue, and maybe Anna can talk more about that. I'll give that to you.
Thank you, Janice. We have like a mutual affection here for each other's projects. So the thing is, James, when they told me I was going to go to teaching online, like tomorrow, and all of my coursework needed to suddenly be accessible to my students who were going through unbelievable things, many of them sharing a hotspot on their phone with their two siblings. This became a very, very different world as an educator. And I can say there was this sort of collective, oh my God, that was coming from the faculty at all these different institutions. And so people started mobilizing all of their resources, which was awesome. But then it was sort of a deluge of riches. Like, how do I kind of navigate this world of so many resources now? Now, they weren't all equally accessible or available. And of course, the natural history collections ones are the very best ones. But how do we create that sort of pathway for educators to learn about these resources and feel comfortable within the classroom and teach them with any sort of fidelity? And so one of the things that Blue has worked towards is this idea of helping the educators get those core skills, and then helping create the sort of materials that allow that to happen a little easier with an easier transition. And so we did sort of a sprint to implementation in the spring, where we did a really quick, let's just help teachers get this in the classroom right now, because they didn't have a semester to develop it. And I can say that there are, there's BCEENET and what they're doing, which is great. There's also iDigBio, that did a similar thing where they galvanized their resources and did a quick iDigBio. And then there's the Cubes Hub, where they have made a centralized set of resources. So educators looking to do something that would be comparable to a lab experience, but online, they sort of curated those resources and pulled them together. So I also see the education community doing a big shift of sharing, similar to the way the community does in natural history collections, kind of learning from each other and then trying to put best practices forward.
And this is Liz again. And one of the other things that happens, our museum has a lot of informal science education. And I know we're talking about undergraduates here, but all of the resources that are being developed for undergraduates then are available for other museum educators to look at and to distill down, and maybe they have to take it another step back. But all that information is there. So there are really some long-term, they're far-reaching knockdown or downstream effects of all of the products that are being developed and put into either modules or entire courses and gathered up on Cubes Hub. As a curator, I can tell my public engagement person, you need to go look here, and you'll get a lot of great examples of how you can engage the public with our specimens. So it really it ends up being kind of a shortcut for us. So there's a lot of very big upsides to this.
Just following up on the informal aspect, we have had a huge amount of interest in participatory work with our collections. There are websites where the public can go and help us transcribe data from images into a structured database. Notes for Nature is one, but there's a whole host of other similar activities. And my institution's very active in putting our data out there because it actually helps us with the transcription. And we can hardly keep up with the demand. And that creates, not only does it do work for us, it gives people an activity that maybe takes them a little bit away from the day-to-day challenges they're facing, but it opens up an opportunity to educate them about things about the collections, how they're used, and we supplement that with stories through a vehicle we call the Hand Lens, which talks about the collections, puts them in context of history, of biology, of world events sometimes. So there are great opportunities here for educating the general public, who hopefully then will be supporters of our collections endeavors.
I'd like to add on to that. I'm Carly Jordan. I'm at the George Washington University, and I'm a co-PI for BCEENET. Sort of back to a question you asked about 10 minutes ago, which was, are these collections useful for educating, people that are going to use the collections, or educating beyond that? And my background is strictly really in science education. I'm very new to the world of collections, but as a science educator, what I see in them is this tremendous opportunity to do lots of really meaningful, active engagement with students, no matter what their major is, right? So the big move in science education over the last 20 years has been about how do we engage undergraduates in real meaningful research, how do we engage them in active learning in the classroom, and now people are starting to think about if we're going to move online, or for instructors that are already operating in this idea of a flipped model where they're asking students to do a lot of things on their own. These resources really provide an opportunity to move courses effectively online, and then if they're not online, to really engage students with good data analysis and quantitative skills, regardless of their major.
Yeah, and that raises a question for me, which is, what kind of work are students, or have students been doing, or will students do using collections? What sort of research is at play there?
We offered a course during the past spring that was on essentially the effects of climate change on Florida plants, and it was all based on using herbarium specimen data, digitized herbarium specimen data. And luckily, we had this set up as a totally computational course. So mid-semester, when everything had to go online, it was a fairly easy transition, even though we were working with freshmen. So the course really emphasized getting students familiar with the collections, what information collections can provide, how you can download the collection information, analyze the collection information, model habitats, and then predict where those habitats are going to exist in the future. And then we could use that information. The students were doing all of this. They were able to make predictions about where certain species, endangered species in particular, might be able to find habitats, suitable habitat in the future. And of course, this taught them some pretty important lessons about the impact of climate change and how we need to translate some of our science into science policy. So there are a lot of ways that we can use these collections data in classes for freshmen through seniors, and obviously into graduate education as well.
So we've been doing this for several years at Central Michigan University in one of our core undergraduate courses. So this is for all majors, not just interested in conservation or organismal biology. So this is where we get the pre-meds. And we give them an opportunity to come up with an open-ended question that utilizes biodiversity data sets, and they can take it wherever they want to go. We don't require it to necessarily be in a specific science area, that it has to incorporate data and certain data skills. And so these students are so much more creative than me and have come up with some just really creative projects. So one group decided they wanted to figure out the best place to camp in Michigan. And so they looked up all sorts of data on the campsites and how many people visited them and where you'd find poison ivy. And we had to talk to them about the fact that just because there isn't a specimen collected from that site doesn't mean it's not there. But then they could look up, well, where are you likely to find poison ivy? And so they did this whole, and they did it with like Boo Boo, who's the Yellowstone Park, I can't remember, like Hey Boo Boo, that one. So they went Yogi Bear. So they did it with like, it was Yogi Bear, and they had picnic baskets, like where were the best restaurants in the state. And so they kind of brought all this in, which was really, to me, a big win, because they were using the data and they were answering this like kind of interesting question. Another one decided to look at where you could find the best tom turkey, the biggest tom turkey in the state, with the highest likelihood of actually getting the turkey. So they had to find out licenses in the state. Then they had to, who's getting licenses in what counties. They also wanted to look to make sure there weren't too many toxins in the water. So they looked at where the toxins might occur. And then they set it up like a Tinder date. So they presented it like a Tinder date, like you're going to swipe left or swipe right or however that works. And I'll admit to not knowing much, but it was a really interesting way of kind of bringing in all these data sets and using them to apply, again, to, I would never have thought of that. So, but that again is us looking at it at the introductory level. What Pam is doing, bringing them into really substantive resource. What I think BCEENET is doing is also like sort of that next step of saying, okay, let's look at how this is incorporated into research, and hopefully making their minds think of new ways of approaching questions.
We did a project at the New York Botanical Garden with high school students, where we had them look at the distribution of rare species. These happen to be carnivorous plants, which are intrinsically interesting. We had them look at the specimens and georeference them, then enter the data into a simple GIS application and calculate the area of the species occurrence and see whether it fell within or outside the threshold of something that would be considered endangered by the International Union for the Conservation of Nature. And then they considered other factors, how old the collections were, maybe the plants aren't there anymore. And then they each gave a presentation about their species, which they had adopted. So the thing is, with the right amount of sort of prepping, you can take these lessons, everything from high school students all the way up to a graduate course.
In this last spring at Widener University, Liz and I worked with students on carrier shells, which are marine gastropods that attach objects, including rocks and other shells and corals and things, to the outside of their own shells. And there's not much known about this group, but there are some really interesting hypotheses about why they do this. And our project did start with a combination of georeferencing and data cleaning and trying to look at the distributions of these. It included some morphological measurements, which then got scrapped with when they sent us all home. But because of the data available, we were able to pivot and do and move it to an entirely online project where the students have found some really interesting information about differences in depths between different species, and they really got a lot out of it. And none of these students they may be upper division, but none of them have plans to become collections professionals. I think we have at least a couple of pre-physical therapy in there, pre-med, and maybe a couple going to graduate school, but it is making those connections with that wider scientific community. And those students, regardless of what career they go into, are going to hopefully remember that these resources are available if there is a question they need to answer.
And I'll just follow up on that, saying this is the second time that we've taught this class, the Widener DMNH class. And the first time around, we had a student who knew he wanted to go on to graduate school, but he didn't know that he wanted to go on to graduate school and include natural history collections. And it was really only after that had happened that he'd had that kind of exposure that all of a sudden a whole new pile of information opened up in front of his eyes. And he thought, well, now wait a minute, I can incorporate this into what I'm doing moving forward. And I think that that's just a terrific outcome.
Absolutely. It sounds like the educational opportunities that are, embedded within this are incredible and, being brought to bear very quickly. I think a last question, I noticed that Joseph Cook's iPhone has joined us as well. So I may address this to him first, but I was hoping we could talk just a little bit about, what the experience has been like, in this era. We've chatted a little bit about it of kind of having to rapidly, switch over to online teaching and stuff like that. But, how has the experience been of, jumping to this online world? And this can be specific to collections, but not necessarily so. I'm trying to get a little bit of a sampling of, various practitioners and researchers, experiences of, how things have been for the past few months. So, very broadly speaking, how's it been going?
Well, I think certain aspects of what museums are doing have progressed very well. Digitization efforts, for sure, I think has given us more time actually to focus on some of the issues that we didn't have time to when we were also taking daily care of collections. But we haven't been in the museum. We're just beginning to open it up, other than checking on temperatures of freezers and so forth for 4 months. And so in some ways there's been an ability to focus on certain things that we didn't have time to before, like even writing proposals. But we are behind now in terms of collection care, for sure.
So I'll respond as well, because I've had experiences from, and I'm sure others have as well, multiple different kind of directions. From the collections perspective, we've also been out for since the middle of March. I guess nobody's been in except to, like you say, check freezers and make sure everything is rained on or anything along those lines. But in our case, the collections seem to have managed without too much trouble. The challenge that we faced as a museum was the pivot to online activities for our education department, and how difficult that was to take what is traditionally a very hands-on, always encouraged to be hands-on, person-to-person interaction, and move that really rapidly onto an online experience. And so that's been one of the bigger issues, I think, at our museum: how to maintain the connection with your community, and with the community of not just biologists but also the local folks. And how do you keep that energy moving? And we've all done, we've been very scrappy about it, and we've been able to do what we can and upload videos and all that. But that's definitely one of the challenges we face.
Well, I just think it's important to recognize that public museums that depend on people coming to them for their bottom line are also struggling financially, and there's been some respite from some of the government programs, but by and large, there's a level of attrition going on as administrators look at budgets and start working on reduction in force plans. And so those are just beginning to show up at some of the major natural history museums and botanical gardens across the country. And, everybody's doing what they can to protect the core mission of the institution. But if you lose too much of the groups of people that make a museum, you're gonna suffer. And so this is a difficult time financially for our institutions. And so we're all hoping we can get back on track, but that's going to depend on a lot of factors, such as when the public feels comfortable about coming out. And so there's a lot of scary financial concerns right now too.
So James, I can speak a little bit to an educational institution, and I have a small collection. My biggest issue there is the people trained to work in the collection graduated. So when I do get to repopulate, I will be training from the ground up versus the overlap that I had planned. But also this is really impacting graduate students and people at these institutions, graduate students who need to be doing research, can't get into the collections or can't get into the field. And so they don't necessarily can't roll it over to another year. I work with a federally listed species that's down to 200 individuals. We can't plan to get out in the field and see if it's still there. So there's a lot that's kind of in the sort of transition phase that seems to be impacting the students a great deal and kind of putting research in a weird place where you have to, I mean, every day I feel like I spend a bunch of energy pivoting to the newest bit of news. Like, okay, we might be able to go out next week. So we're arranging vehicles and hotels, and then now we can't, or we can go, but only one person can go in a vehicle. And so that just doubled our travel costs. So there's just a lot of uncertainty in the science research world as well.
I think the travel restrictions and the fact that we don't all want to be sitting in rooms together at conferences is all having a big impact as well. And, you know, we were discussing earlier some of the benefits of the online conferences, but one of the problems that we see is that societies also tend to not be able to generate some of the revenue that helps support their programming throughout the rest of the year. And so we're seeing, particularly as organizations are maybe not charging registration, they're not making any money, and yet we want to be supporting our graduate students in research and in field work and all of these sorts of things into the future. So the support for professional organizations, I think, is something that we'll just have to be very careful and monitor over the next few years.
Yeah, this year for the Digital Data Conference, we decided that if you wanted to come, you could come. You can register. If you'd like to make a contribution, you can come with a registration fee, and that contribution goes to sustaining the conference as an in-person conference in future years. And I was pretty amazed. We have a sort of a bank with NSCA and AIBS. Our money goes there that we can use later so that we can build a sustainable conference. And it turns out that we raised over $11,000 from people who just paid because they said, yeah, I want to contribute to keeping things going. And I thought, well, that's about 50% of what we normally raise for people when it's an in-person conference. Because I think we had more people coming, but not actually coming. They were virtual, and they wanted to participate in helping support the notion. And so, maybe we can get to a point if we, don't make it free. Say, folks, we've got to keep this going somehow. There are a lot of people putting time into it, and how can we keep it going? And out of your altruism and the goodness of your heart, help us out here.
Yeah, absolutely. And it's certainly an important moment to support the institutions and organizations that you find valuable, because I think, as we've discussed today, they may have roles to play in, very important things in our lives. So a strong case for advocacy, for funding, and for, doing everything that we can ourselves. On that note, I would like to thank you all very much for joining me today. I think this has been a successful podcast. We've only scratched the surface on many of the topics, of course, but I think what we have covered, we've covered very well, and I appreciate your time and your chatting with me today. So thank you.
Thank you very much.
Thank you.
It's been a lot of fun. Thank you.
That was great.
Thank you.
Thanks, James.
Bye.
And that concludes this episode of BioScience Talks. Just a reminder, the journal BioScience is published by Oxford University Press on behalf of the American Institute of Biological Sciences and is made possible by the support of our members and donors. Thank you, and talk to you next time.